Hi there. Let's chat about the amazing world of living things, past, present, and maybe even beyond Earth. Scientists used to sort all living things into six big groups: Animalia, Plantae, fungi, Protista, Archaea, and bacteria. Kind of like labeling different types of foods in your kitchen cupboard, organizing, but maybe not telling the whole story. Everything revolved around a few checklist items, such as a nucleus versus no nucleus, membrane bound organelles versus no organelles, no membrane bound organelles, cell wall thickness versus different types of them, like fences make good neighbors, multicellularity, and how each organism eats. Fast forward to the mid 1970s, scientists compared small subunit ribosomal RNA, so-called SSU RNA. It's like they were matching up the genetic barcodes of different organisms. And surprise, surprise, they discovered that living things actually split into the three main categories, the three main domains we have today: the bacteria, archaea, and eukarya. Bacteria and archaea may both be prokaryotes, which means they have no nucleus and no true membrane bound organelles. But genetically and in terms of their membranes, they are as different from each other as they are from the eukarya, the domain that includes us animals, plants, and fungi. Prokaryotes showed up on Earth about 3.5 million years ago, long before your favorite houseplant or your pet iguana. They lived in every nook and cranny imaginable. Got a boiling hot spring? They're in there. Deep freeze in the Antarctica? Also there. Inside your gut? Yep, in higher numbers than your own cells. Makes you wonder who's really running the show, right? They're also nature's recyclers, tossing carbon, nitrogen, and other essentials back into the mix so new ecosystems can form. Definitely something to think about when you're composting your veggie scraps next time. Once we began sequencing DNA, we realized prokaryotes were far more diverse than we ever guessed. Imagine rummaging through an old attic and finding boxes of hidden treasures. DNA sequences basically opened up that attic for microorganisms. Discovering those differences forced us to rethink classifications, reminding us that we're still learning who we are in this grand scheme of life on Earth. Earth is about 4.5 billion years old, give or take. This number pops up from meteorite dating, which is kind of like checking the born on date for the solar system. Because see, you couldn't date how old Earth is with its own rocks, since they are created after Earth itself got created. The early atmosphere is anoxic, which means it has no oxygen. So only anaerobic organisms could party in there. Those are the ones who can live without the oxygen. The earliest organisms used hydrothermal vents, which are fissures in the Earth's surface, to obtain energy. About 3 billion years ago, the evolution of photosynthesis created phototrophs, which are organisms that can use sunlight for energy. Then along came cyanobacteria, which we know as blue-green algae. They use sunlight to create energy, but also they gobble up carbon dioxide and then pumping oxygen into the atmosphere, and that paved the way for all us oxygen lovers. See here, you can see how the cyanobacteria turn a stream at the Yellowstone National Park green. Before our comfy oxygen-rich environments took shape, the planet was basically bombarded by radiation and had volcanic eruptions galore. So the first prokaryotes likely thrived deep in ocean vents and underground, like cozy basement dwellers. Over time, a bunch of microorganisms stuck in a gooey matrix of polysaccharides plus proteins and nucleic acids. We call that entity a biofilm. A large, thick collections of biofilms then created biomicrobial mats. Microbial mats are multi-layered sheets of prokaryotes that are formed on most surfaces. And these mats can range from a few millimeters up to a few centimeters in thickness. They're basically living carpets of bacteria on the archaea held together by a gummy gooey-like substance. Talk about group living at its finest. See here, we can see a microbial mat at a hydrothermal vent in the Pacific Ocean. The arrow points to a chimney which lets its gases escape. So a biofilm is a community of microorganisms that adhere to a surface and are encased in a self-produced slimy extracellular matrix. And a microbial mat is a multi-layer sheets of these biofilms and create these structures, larger structures. Biofilms show up all over the place, on industrial surfaces, in your kitchen sink, on cutting boards, and even on toilets. They are also good at clogging pipes. Their super team effort makes them tough to destroy, way stronger than if they were free living, or also known as planktonic prokaryotes. Microbial mats got fossilized, forming structures called stromatolites. They are like layered rock pancakes. These fossils date back about 3.5 billion years. Knowing this helps us look for similar signs on other planets as well. Because if microbial mats are formed here, who said something like that couldn't be formed on Mars or beyond? So you see those stromatolites here, and they are circa about, these ones here are circa about 1.5 million years old and are found in the Glacier National Park in Montana. Pretty looking things. Some bacteria and archaea live in places that will make the rest of us shrivel up and cry. Super hot vents, acid pools, deep ice, high radiation zones, you name it. We call them extremophiles, or lovers of extremes. They might help us discover new medicines or industrial processes, like specialized enzymes, for example. Plus, they open a door to the idea that life in our solar system might exist in the weirdest corners. Looking up at you, Mars. Ironically, many of these extremophiles can survive in milder environments. All right, so next, let's look at some characteristics of prokaryotes.